I'm Dr. Ahmad Haidari. I'm the course leader for MSc Molecular Biology and Health at Staffordshire University. Our MSc course is in part-time and full-time for distance learning cohort and full-time only for on-campus face-to-face cohort. The course is designed to study the structure and function of biological molecules within the cells, the tissue and organ system in relation to health and disease condition. This course is designed for those students who have the passion for understanding the fundamental mechanism of life, health, and variety of disease conditions. The course is composed of 8 individual modules. These individual modules are delivered in 3 semesters. In the semester 1, the student will study the principle of molecular biology, the molecular biology of a disease, and the bioinformatics. These modules help students to explore the structure and function of the genes, genetic sequencing, and also genetic variation in relation to health and disease condition. In the second semester, the student will study practical skills in molecular biology, molecular genetics, and project proposals. These modules help students to gain hand-on experience in cutting-edge laboratory techniques, such as quantitative PCR and DNA sequencing technology and gene expression analysis. And on the third semester, the 2 last modules would be the current advances in molecular biology and research project. In the current advances module, the student will focus on the core principle and application of gene editing technology in the context of diagnosis, prognosis, and disease condition. And the research project module allows students to undertake a real-life scientific research project. This project helps students to work independently in the lab and develop their advanced laboratory skills. This screen can show you the percentage of cells are gated or healthy at the early stage of apoptosis or programmed cell death and the late stage of programmed cell death, and the cells are now breaking down in pieces called necrotic cell death. This machine, as you probably be familiar with, is quite expensive. It's about £100,000. So we use this for teaching and also for research project. So a student will use this during the practicals when they're assessing the effect of different types of compound when they treat the cancer cells and normal cells with cytotoxic drugs and affect the cytotoxicity of compound to measure how the compound interact with the cells. Either the compound is effective cytotoxic enough to degrade cancer cells. So this type of measurement can be assessed by this machine. Apart from that, we can use this machine for cell cycle analysis. For the cell cycle analysis, as you know, most of the cancer cells our student, MSc student, or work with, we measure the cell proliferation rate of the cancer cells. So we can use the cell cycle analysis to find out the doubling time of the cancer cells and then compare that with normal cells. So we've got a normal batch of breast cell line, and we've got a cancerous version of the breast cells. Then we run both panels using this machine, and then we can identify the proliferation rate of the normal cell and the cancer cell, and then we can identify the amount of the biological molecule expressed in each stage of the cell cycle can be quantified by this machine. This instrument, as you can see, is a fluorescent microscope. Fluorescent microscope is used to identify, to visualize the morphology of cells, the morphology of normal cells, the morphology of cancer cells, and the morphology of inflammatory cells. So this instrument is used to stain the specific property of cells, whether the property is a carbohydrate or is a protein or is a nucleic acid, can be stained with specific dye and then can be observed by this instrument. So this machine is very important. Comparing with the fax machine I discussed here, the fluorescent microscope helps us to identify the anatomical location of biological molecule within the cell, or if it's the plasma membrane. It's very important diagnostic, has got diagnostic value. When we use treating the cells with different type of compound, we would like to observe if the compound are managed to enter the cells or still are attached to the cytoplasmic membrane of the cells, or if it's inside the cell, if it's in the cytoplasm or attached to the plasma, the nuclear membrane, or if entered into the nucleus of the target cells. This instrument is called seahorse. The seahorse technology help us to identify the metabolic activity of cells when they are treated with different type of compounds. If cells are at the higher metabolic rate, or at the normal, or in the disease condition, or the cells are treated with different types of compound, how they behave. So the metabolic activity of cells, specifically cancer cells, are assessed with this instrument. If you ask me the question how this course can manage to provide a wide ranges of career opportunity, I would like to answer the question. Look at the structure of our MSc course. If you look at the structure of MSc course, you can see the course is at the intersection of biology, chemistry, and bioinformatics. Therefore, the degree you will achieve from this course would be a versatile degree. So that can open a wide range of career opportunity for you after graduation of this course.